This is part 4 of the cell division lectures, and we are finishing up with the section on mitosis, um, or the division of the regular body cells, and we will be moving into meiosis, which is the division of sex cells and the formation of egg and sperm. But we had left off discussing how mitosis is related to cancer. Uncontrolled mitosis can lead to cancer. And what can what can cause the cancer to begin with is a mutated gene that leads to a mutated protein. P53 normally reacts this way. If you look at this chart here on the left, DNA damage, the cell cycle abnormalities, and hypoxia, meaning low oxygen, not enough oxygen getting into the cell. Any damage in the DNA, any abnormalities within the cell cycle, and low oxygen will cause P53 to either undergo apoptosis directly, which is programmed cell death. That means it's genetically programmed that the cell will just destroy itself, or the cell cycle will go into arrest, which means it will pause. The DNA will repair itself, and the cell cycle will restart. Now, if the cell cycle goes into arrest and the DNA is not able to repair itself, then of course it will lead to apoptosis that way. So when cellular damage occurs down here at the bottom, P53 arrests the cell cycle until the damage is repaired. If the damage can't be repaired, then apoptosis occurs, and that's programmed cell death. So that cell cannot divide and become a tumor. Now, in a mutated P53, the cell damage, the cell cycle abnormalities, and the hypoxia in the mutated P53 do not cause apoptosis in the cell. They cause the cell cycle to continue, and the cells that are dividing that we don't want to divide can become cancerous. They can become a tumor, and cells from that tumor can spread. And that's what cancer is a tumor that has spread, and we call that metastasis, or we say that it has metastasized. Now, moving on, we're going to talk about cell division as it relates to sexual reproduction. Sexual reproduction, specifically meiosis and fertilization. So meiosis is the formation of the sex cells, and fertilization is the joining of the sex cells. Formation of egg and sperm, and then joining of egg and sperm. That's the definition of sexual reproduction. And what it does is it introduces variation, genetic variation, into the offspring. The vast majority of eukaryotic organisms, both multicellular and unicellular, can or must employ some form of meiosis and fertilization to reproduce. And that includes plants, that includes animals, that includes fungi. All right, so the differences. They're both forms of nuclear division within the cell. Meiosis has the same stages as mitosis, but instead of the parent cell, in other words, in humans, has 46 chromosomes. But at the end, first of all, mitosis is 2 divisions of the parent cell. So you have 2 daughter cells that form during the first division, and their chromosome number is halved. Then the 2 daughter cells divide again, basically clone themselves. So you end up with 4 daughter cells that are haploid, that have only 23 chromosomes. So the starting nucleus is diploid, and the nucleus that results at the end of meiosis is haploid. So you start with diploid and you end up with haploid. In mitosis, it was diploid to diploid. To achieve this reduction in chromosome number, meiosis consists of 1 round of chromosome duplication, just like mitosis, and 2 rounds of nuclear division. And this is how it works. We start out with an S phase, which is when the DNA is replicated. So it's always replicated at the beginning of meiosis. And then we have 2 divisions. So the first prophase is called prophase 1. In prophase 1, the same things happen as in prophase of mitosis. The nuclear envelope and the nucleolus disappear. The spindle begins to form. The chromatin condenses into chromosomes. But we have to add one more thing, and that is that the homologous pairs match up, and that process is called synapsis. We'll discuss this more in a little bit, but for right now, I just want to tell you, in prophase 1, the homologous pairs match up, and that's called synapsis. And then a process called crossing over, where some of the chromosomes from the homologous pair can actually exchange places, that occurs during prophase 1. Then we have prometaphase 1, which is pretty much the same as metaphase 1. The spindle fibers or spindle microtubules attach to the kinetochores of the chromosomes. In metaphase 1, the chromosomes are lined up, but this time they're lined up in pairs. Homologous pairs line up rather than individual duplicated chromosomes. So you see a pair of chromosomes lining up with other pairs of chromosomes instead of just lining up kind of singly or single file. In anaphase 1, instead of the sister chromatids being pulled apart, the homologous pairs are pulled apart. And then telophase 1 and cytokinesis are fairly similar as what you would expect, but sometimes cytokinesis does not occur after the first division. It doesn't occur until the last division. Then we have prophase 2, and all of the second stage prophase 2, prometaphase 2, metaphase 2, anaphase 2, all of those stages are exactly the same as mitosis. So in prophase 2, the chromosomes condense, the nucleus disappears, and all that stuff. The same thing that happens in prophase of mitosis. In prometaphase, the spindle microtubules attach to the kinetochores of the sister chromatids. In metaphase 2, the sister chromatids line up at the center of the cell, and in anaphase 2, the sister chromatids pull apart. And then in telophase 2 and cytokinesis, you end up with 4 daughter cells. The original parent cell was diploid. It had 46 chromosomes. By the end of meiosis, the 4 daughter cells each have the haploid number of chromosomes, which is 23. Okay, and so early in prophase 1, homologous chromosomes come together to form a synapse. That's the homologous chromosomes, the one from the mom and the dad, they're homologous chromosomes when they're the same size, and they have genes for the same traits. Let's say, for example, that the mom is the red chromosome and the chromosome that came from the dad is the blue chromosome. Well, they both have genes for the exact same traits, but remember, they don't have to be the same versions of the genes. So if this, for example, was one of the skin color genes on the mom, you would have the same gene here. It would be for skin color, but they don't have to be the same version. One can be darker, one can be lighter, you know. So they contain genes for the same traits. They're homologous. They're pairs, but they don't have to have the exact same versions of those traits. But the homologous pairs match up. That's called synapse. And then a process called crossing over occurs. And that is when we said the dad was going to be the blue chromosome and the mom was going to be the red chromosome. Well, his genes are big A, big B, big C. And the mom's are little a, little b, little c. But with crossing over, you can get different combinations of genes. You can get big A, big B, little c. You can also get little a, little b, big C, which you would not be able to get that much variety without crossing over. So crossing over, what it really does is it increases genetic diversity. In prometaphase 1, same thing as what you expect is that the microtubules from the spindle attach to the kinetochores of the homologous pairs. Metaphase 1, the homologous pairs are lined up in the center of the cell, and they assort themselves randomly in metaphase 1. So you can have, for example, here you've got 2 chromosomes from the father on the left and 2 from the mother on the right. Here you've got on the left 1 from the father, 1 from the mother, and on the right 1 from the mother, 1 from the father. In other words, how they line up, how they pair up is random. It's a independent or assortment or random assortment during metaphase 1. And it's important for the same reason that crossing over is important. It increases genetic variation in the offspring. This is how we get so many differences that sometimes you can have a brother and a sister, a biological brother and sister that look no more alike than 2 strangers. Most of the time they will have similarities, but sometimes I know you've seen people in some families that don't look like they came from the same family. And that is because genetic variation occurs during the formation of sperm and egg. And sometimes that genetic variation is really makes the offspring really different from each other. So in anaphase 1, the sister chromatids stay together, but the homologous pairs are pulled apart. The homologous chromosome pairs are pulled apart. And you can see that here. So this is what happens in prometaphase 1 and then anaphase 1, is that here's the homologous pairs. The pairs are pulled apart in anaphase 1. But in prometaphase 2, you just have this one chromosome consisting of 2 sister chromatids, and those sister chromatids are pulled apart just like they are in anaphase of mitosis. And then telophase 1 and cytokinesis are very similar. Sometimes they don't occur like all the way, that they don't go all the way through the process to where you end up with 2 separate daughter cells. You know, sometimes they don't separate. They don't separate fully until the end of meiosis 2. So what happens is sometimes there's not an interphase, but sometimes there is an interkinesis or a break before entering meiosis 2. And interkinesis does not have an S phase. So the DNA does not duplicate again between meiosis 1 and meiosis 2. It only duplicates before meiosis begins. So meiosis 2 is similar to mitosis. And so all of these stages, I'm not going to go through this again for time, but all of these stages are identical to the same stage in mitosis. Prophase 2, the chromosomes are condensed. Prometaphase 2, the spindle fibers attach to the sister chromatids at the kinetochore. Metaphase 2, the sister chromatids are lined up at the equator of the cell. Anaphase 2, sister chromatids are pulled apart. And telophase 2 and cytokinesis results with 4 daughter cells that are each haploid, that contain half the number of chromosomes as the parents. All right, so sexual reproduction. Nearly all eukaryotes undergo sexual reproduction. The variation introduced into the reproductive cells by meiosis appears to be one of the advantages of sexual reproduction that has made it so successful. Meiosis and fertilization alternate in sexual life cycles. The process of meiosis produces unique reproductive cells called gametes.